Hello everyone, I am Vinci, Assistant Professor, Department of Botany, Krebs College, Nagarkoil. Now I am going to discuss the topic Krebs cycle. The oxidation of pyruvic acid into carbon dioxide and water is called Krebs cycle. It was discovered by Krebs in 1936. This cycle is also called citric acid cycle because the cycle begins with the formation of citric acid. The citric acid is a carboxylic acid containing 3 COOH group. Hence, this cycle is also called tricarboxylic acid cycle. The entire Krebs cycle occurs inside the mitochondria. It is an anaerobic process. Krebs cycle involves 10 steps. First step formation of acetyl-CoA, second step formation of citric acid, third step dehydration, fourth step hydration, fifth step dehydrogenation and decarboxylation. Step oxidative decarboxylation, seventh step hydrolysis, step 8 oxidation, step 9 hydration and step 10 dehydrogenation. Step 1 formation of acetyl-CoA. The raw material for citric acid cycle is acetyl-CoA. It is formed from fatty acid by beta oxidation and from pyruvic acid by oxidative decarboxylation. Pyruvic acid combined with CoA to form acetyl-CoA. This reaction is catalyzed by set of enzyme called pyruvic acid dehydrogenase. In this reaction, two hydrogen atom and one carbon dioxide molecule are removed. Hence, this reaction is called oxidative decarboxylation. The two hydrogen atom are accepted by NAD plus and NAD plus is converted into NADH. This reaction occurs outside the mitochondria. Step 2 formation of citric acid. Acetyl-CoA transfer its acetyl group to oxaloacetic acid to produce a 6-carbon citric acid. This reaction is catalyzed by the enzyme citric acid synthetase, which is the release of CoA. Citric acid undergoes dehydration with the help of the enzyme acanitase to form 6-aconic acid. It remains bound to the enzyme. 6-aconic acid undergoes hydration with the help of the enzyme acanitase to form isocitric acid. 5. Dehydrogenation and decarboxylation. Isocitric acid is dehydrogenated to alpha-beta-glutaric acid and carbon dioxide by isocitric acid dehydrogenase enzyme. The electron is upsetted by NAD plus to form NADH and H plus. In this step, oxidative decarboxylation occurs in alpha-beta-glutaric acid to form succinyl-CoA by the addition of CoA. This reaction is catalyzed by the enzyme alpha-beta-glutaric acid dehydrogenase with the help of NAD+. Hydrolysis. Succinyl-CoA is hydrolyzed to succinic acid by the enzyme succinyl diokinase by releasing CoA. A phosphorylating enzyme is involved during the course of the reaction. Guanosine diphosphate react with inorganic phosphate to form guanosine triphosphate by the process known as substrate phosphorylation. This GTP can enzymatically react with the ADP to produce ATP and GDP. Oxidation and the succinic acid is oxidized to fumaric acid. The enzyme involved is succinic acid dehydrogenase with the help of flavin adenine dinucleotide. Hydration with the addition of one molecule of water in the presence of enzyme fumarase, fumaric acid is converted into malic acid. In the last step, under the influence of enzyme malic acid dehydrogenase, malic acid is converted back to oxaloacetic acid. Dehydrogenation was achieved with the help of NAD+. Krebs cycle summary. Krebs cycle occurs in the mitochondrial matrix acetyl-CoA, which is produced from the end product of glycolysis. Each citric acid cycle form the following product: 2 molecules of carbon dioxide released, removal of carbon dioxide or decarboxylation of citric acid take place at 2 molecules. In the conversion of isocitrate to alpha ketoglutarate, in the conversion of alpha ketoglutarate to succinyl-CoA. 1 ATP is produced in the conversion of succinyl-CoA to succinate. 3 NAD plus are reduced to NADH and 1 FAD is converted to FADH2. 2 molecules of acetyl-CoA are produced from oxidative decarboxylation of 2-pyruvate. So 2 cycles are required per glucose molecule. And to summarize, for complete oxidation of a glucose molecule, Krebs cycle yield 4 carbon dioxide, 6 NADH, 2 NFADH2 and 2 ATP molecule. Each molecule of NADH can form 2-3 ATPs and each FADH introduces 2 ATP and oxidation in the electron transport chain.